क्या कर रहे हो? तूने होमवर्क किया? देखा? अभी कौन सा लेक्चर है? मैथ्स में किया दोनों ने होमवर्क? आ रहा है बेर को सब कुछ? कब है एग्जाम? पता नहीं, अभी तक अनाउंस नहीं किया। अच्छा, और तूने वो आफ्टरनून को प्रैक्टिस किया मैथ्स के सम का? आ रहा है सब कुछ? इंग्लिश गया ना टेक्निकल भाषा में, हो गया? इसमें क्या है? तुम्ही एक ही स्कूल केलं ना तो होता और ये तू लास्ट टाइम हंसा कब था तू हंसता नहीं है तेरा टॉपर तो है स्कूल में एवं हाँ वो कहाँ जाता है क्लास के लिए ऐसे होते स्वीट मुझे कितना मिला था इसमें इसका बताना ये करता है ना बराबर सम तेरा कॉपी नहीं करता है ना विषय है ना इस पे ध्यान देना ये होमवर्क नहीं करता है ना विषय करता हूँ चलो बाय What did we finish, Ramesh? Which cell organelle we were discussing last time? What was it? Golgi bodies we were discussing. How far did you draw the diagram? Show me. The diagram, how far you finished? Again, you label with pen. And also this way, anyhow you label the diagram. What about you? Show your diagram. This is better. This is better. Always to label diagram with a pencil. Always to label diagram with a pencil. You will not label the diagram. You will redraw the diagram again. Go to the next page. Take a pencil. Draw there. Take a pencil, and as I draw, you will draw the diagram. We are almost done with the cell organelles of the animal cell. I think we are talking about the Golgi bodies. Let us continue. Which are the organelles we are done? Tell me. Which organelles? Plasma membrane have we done? Plasma membrane we are done. Then after the concept of plasma membrane, what? Cytoplasm. Done. Cell wall. Done. Mitochondria. Golgi, endoplasmic reticulum, we must have finished, no? Then Golgi. Lysosomes? No. So we have to start with Golgi. So now Golgi complexes, if I talk about Golgi, now say suppose this is the nuclear membrane. The nuclear membrane, we are going to talk about the nucleus also today. This is the nuclear membrane. Talking about the endoplasmic reticulum, how many types of endoplasmic reticulum? 2 types. What is the difference? What are the names? Rough endoplasmic reticulum and smooth endoplasmic reticulum. What is the major difference between the rough and the smooth? Rough endoplasmic reticulum is consisting of ribosomes on the outer surface. Smooth endoplasmic reticulum consists of ribosomes or without ribosomes? Without. What is the function of the rough endoplasmic reticulum? Function of the rough is protein synthesis, because ribosome's function is protein synthesis. What is the function of the smooth endoplasmic reticulum? Did we write? Formation of lipids and fats. No? Formation of lipids and fats. The function of the smooth endoplasmic reticulum is lipids and fats. So that you have to remember. Function of the smooth endoplasmic reticulum. If we are talking about the Golgi, where is the Golgi placed and how is the Golgi placed? The Golgi is always like this. In the form of some disc-like structures, in the form of some wheel-like structures. And the Golgi is always the site where a large number of enzymes and proteins are produced. The Golgi would always be producing certain small vesicles like this, small vesicles like this, always coming out from the Golgi. But one type of vesicle we may come across which is containing some digestive enzymes. Did we talk about the functions of Golgi? No? Yes? The functions of Golgi. So if I have to talk about what are these Golgi complexes made up of, let me start with the Golgi again. So I talk about the Golgi complexes. The Golgi complexes. Now what are these Golgi complexes made up of? Tubular structures, satellite structures. And what are the tubular structures and satellite structures of the Golgi known as cisternae? What are cisternae, tubular or satellite structures of the Golgi? Now this Golgi produces a variety of enzymes. What is their function? Produces a whole lot of enzymes. They produce variety of enzymes. A whole lot of enzymes. But now that I said it produces a whole lot of enzymes, then what are the other functions of it? Let me finish with the Golgi. Then what is the other function of the Golgi complex? They would help in transportation of various cell secretions. Various cell secretions from one cell to the other. So now, when I talk about the functions of the Golgi, what is the Golgi doing? The Golgi is always involved in producing variety of enzymes. 40 different type of enzymes the Golgi can produce. Now this Golgi can produce 40 different types of enzymes. And now that we are talking about that the Golgi produces 40 different types of enzymes, how are these enzymes carried? Always in the form of small vesicles like this. Always in the form of small vesicles like this. And in the form of these vesicles, now this can be carried from one part of the cell to the other. And sometimes they will also be moving from one cell to the other, always like this. So that's why I have written on the second point. What is the other function of the Golgi? The other function of the Golgi is transportation. Transportation of the secretions of the cell. Transportation of the cell secretion from one part of the cell to the other or from one cell to the other. Clear? But now, with the reference of Golgi, I may understand that the Golgi produces vesicles carrying digestive enzymes. The Golgi produces vesicles and one such vesicles carry digestive enzymes. What are digestive enzymes also known as? They are also known as hydrolytic enzymes. Something like I may talk about that maybe this type of vesicles. These vesicles I would name them as lysosome. Why? These are the vesicles with hydrolytic enzymes. These are the vesicles with hydrolytic enzymes. How will I label this part as? What is the name of this part of the cell? This organelle of the cell is the Golgi complexes. This is the Golgi complex. And what is that vesicle from the Golgi which contains digestive enzyme known as lysosomes? So I'm saying is that whatever type of vesicles Golgi produces so many types of vesicles. Every vesicle is known as lysosome. Only the type of vesicle which contains digestive enzyme. So that is only one type of a vesicle which contains digestive enzyme. One type of vesicle that contains digestive enzymes or hydrolytic enzymes. Vesicles carrying digestive enzymes or hydrolytic enzymes, what are they known as? These are known as lysosomes. So, can we say that lysosomes are originated from the Golgi? Yes. Golgi gives rise to lysosomes also. Golgi gives rise to lysosomes. Now we try understanding some features, functions of the lysosomes. And what is the definition of lysosomes? What are lysosomes? Vesicles formed from Golgi containing digestive enzymes. Okay? Now, trying to understand some functions of the lysosomes. What are some functions of the lysosomes then? Some functions of the lysosomes would be, number one, two functions you have to study. Number one, I would say they help in killing the microbes. They help in killing the microbes, unwanted pathogens that enter the cell. Unwanted pathogens that enter the cell. Okay. So they would be killing the microbes or unwanted pathogens that enter the cell. That is how the lysosomes also prevent us from any type of diseases. Microbes entering the cell, lysosomal enzymes will break them, digest them. So we will not get disease. So killing of the microbes and unwanted germs, pathogens inside the cell is carried out by the lysosomal enzymes. But what is something more? Something more is when a cell is old or worn out, meaning dead, not functional anymore. When a cell is dead or worn out, the lysosomal enzymes will dissolve the cell parts. And will be thrown out of the body as waste substances. With this respect, the lysosome is also known as what? Suicidal glands. What is the meaning of suicidal glands? When is the lysosome known as suicidal glands? The lysosomes are known as suicidal glands only when they can kill or destroy the dead or worn out cells. So what is happening? The lysosome is known as the suicidal glands only where the lysosomal enzymes can dissolve the cell membranes and organelles and every part of the cell. So when we are talking about the functions of the lysosome otherwise always will kill the germs, pathogens, unwanted substances entering the cell. But if the cell is a dead one, then the lysosome will go and fuse with the nucleus, fuse with the mitochondria, fuse with the Golgi, destroy, dissolve all the membranes, destroy the cell, which will be released out as waste substances from our body. So, every day, hundreds of cells inside our body die out. Are we aware? Is there any separate cleaning process we have to do for ourselves? Nothing. These lysosomal enzymes can constantly dissolve the cells, throw the waste substances into the blood, and they get filtered out along with the waste substances, the function of the kidney. So this is the regular process of throwing out of the waste substances that happens. So the Golgi and the lysosome, how are they related? You should understand. Next is, after the Golgi, the mitochondria, the lysosome, next is we will be talking about the nucleus. Now let us talk about the parts, functions of the nucleus, and after that I will give you some questions. The nucleus, plant and animal cell both possesses the nucleus. If it is an animal cell, the nucleus will always be occupying almost the central portion, the central position. If it is an animal cell, the nucleus is always in the center. What are the parts? The nucleus will always have a double-walled membrane like this. What is this known as? This is known as the nuclear membrane. This is a eukaryotic cell, so definitely it will be having a well-developed nuclear membrane. The nuclear membrane is always double-walled. Inside this, there is a cytoplasm matrix. What is this matrix known as? Nucleoplasm. Then next one, there will be a large number of thread-like structures. What are these thread-like structures known as? This will be known as chromatin fibers. These are known as chromatin fibers. Next, at one end of the cell, there is a extensively dark, dense region. What is this dark, dense region known as? This is known as nucleolus. What else? There will be some opening or gaps at the nuclear membrane known as the nuclear core. So that is the entire structure of the nucleus when I talk about the eukaryotic cell. The eukaryotic cell has this type of a structure, and this is what is the nucleus comprising of. Talking about some other characteristics of the nucleus, the nucleus would generally have some other specialized characteristics. I talk about the chromatin fibers or the chromatin threads. Now, talking about these chromatin threads, I may understand that these are the coiled and the long structures like this. The long coiled structures like this. I want both of you to draw the nuclear part complete in your diagram and label all the parts nice. I'll give you 2 minutes. Complete this one. Done? Okay, now the chromatin threads. What is present inside the nucleus? Fine long thread-like structures, chromatin threads. But these chromatin threads, I get to see that these chromatin threads at times, they become extensively coiled. They become extensively coiled like this, and this is what is known as chromosomes. So what are chromosomes? Chromosomes are extensively coiled chromatin threads. What is the difference between chromatin thread and chromosome? When the thread is fine, long, thin, chromatin threads. When they are extensively coiled, chromosome. Okay. But now to understand that what type of structures are present inside the chromosome, what is there inside the chromosome, we get to see that inside the chromosome they have got some ribbon-shaped structures like this, which is almost flat, extensively coiled, curved. So what is inside this chromosome? Inside the chromosome there are such type of ribbon-like structures, and what is this ribbon-like structure known as? DNA. What is the full form of DNA? Deoxyribonucleic acid. You should remember this name, deoxyribonucleic acid. Clear? So I told you what are chromatin, what is chromosome, and what is the chromosome made up of? Chromosome consists of a ribbon-like structure known as the DNA, and the DNA full form you have to remember. But now in the DNA I get to see that there are some specific regions in the DNA, and every region in the DNA is responsible for one characteristic that we show. Maybe I talk about the height, I talk about the eye color, I talk about the skin tone, any type of a characteristic that we can think of ourselves is all in our DNA. So the DNA contains a large number of such type of sections, small segments, and these segments are regulating every characteristic that we show: the curly hair, the shape of the nose, the shape of the finger, the height, the skin tone, etc., etc. Then what are these regions of the DNA known as? Segments of the DNA known as? What are these segments of the DNA known as? These segments of the DNA are known as genes. What are genes? The segments of the DNA that regulate a large number of characteristics that we show. Clear? So these chromosomes and the chromatin that we are talking about, what is the function of the nucleus? Let us try to understand. What is the nucleus doing? I would say that these chromosomes, they are the hereditary material. Why are they known as the hereditary material? Because they carry genetic information from the parents to the next generation of offsprings. So why are the chromosomes known as the heredity carriers of heredity? We would say chromosomes are the carriers of heredity. Why chromosomes are known as the carriers of heredity? Because they carry information from one generation to the other. How? When the chromosomes, I would understand how would the chromosomes carry hereditary material? If you remember, in the 8th standard, a little bit you have studied about plant reproduction. You have studied, remember, where you had come across that the male cells containing a number of chromosomes, female cells containing a number of chromosomes, results in the formation of a zygote. And that zygote will carry chromosomes of the father, chromosomes of the mother, not only for an animal, for a plant also, the male part, female part. And then the zygote is going to give rise to a new organism. Yes or no? So that is how we are talking about that, how characteristics of chromatin material passes on from one generation to another. That is the reason the chromosomes are known as the hereditary material. What is the meaning? Meaning is they pass on information from one generation to another, parents to offspring. So what is the general function of the nucleus? What are you supposed to write? General functions of the nucleus. Number one, nucleus regulates all the functions of the cell. Nucleus regulates, you have your textbook? Textbook? Why not? Textbook you have to bring to class every day. Textbook. Selena, school textbook you'll have to bring to class every day. Because all these things, functions you did not write. What is very important, that's it. I'll ask you to mark from the textbook itself. So when we are talking about what is the function, number one, what would be one specific function of the nucleus? Regulates the functions of the cell. What is one specific function of the nucleus? Regulates all the cell functions. Regulates all the cell functions. What type of enzymes the Golgi should produce? What type of energy molecule? How many energy molecule mitochondria should produce? Ribosomes should produce? Exact everything. What else? Now that I'm talking about the nucleus also plays an important role in the process of cell division, or regulates the process of cell division. So the nucleus also regulates the process of cell division. Regulates cell division, meaning what? One cell will give rise to two cells. So the process of cell division is a function of the nucleus and regulates the other functions of the cell. Regulates the functions of the cell organelles. All these are functions of the nucleus. Okay, now next is, after the nucleus, one more function, one more organelle we'll have to talk about, that is the centrosome and the centriole. Let us just finish with this. Centrosome, centriole. Where is the centrosome, centriole located? The centrosome, centriole is almost like an organelle here like this at one pole of the cell. Pole of the cell. This part of the cell is known as the pole of the cell. And how many poles does a cell have? 1, 2. 2 ends of the nucleus. And then this organelle will always be having a structure something like this. This itself is a cell organelle. What are the parts? If I talk about what is the labeling of this one, this is known as the esters or the estral rays. What about this one? This is known as the centrosome. And what about this one? This is known as the centriole. This is known as centriole. So the centrosome and the centriole, remember the centrosome and centriole that I am talking about is only present in the animal cells. Do we see this organelle ever in the plant cells? No. We don't have this centrosome, centriole in the plant cells ever. So, talking about the centrosome, what is this centrosome consisting of? I have just drawn the structure there. The centrosome, the structure would be something like this, a small amount of cytoplasm and 2 protein bars like this, 2 protein units like this, surrounded by some rays of proteins. That is the entire cell organelle. Okay. So now talking about this entire cell organelle, that this particular cell organelle centrosome, what is it consisting of? It consists of a specific type of a protein. How many proteins? It has got 2 proteins. And what is the type of the protein known as? The proteins are known as microtubules. Microtubular protein. This is known as microtubules. So this type of proteins, the type of protein that is made up of 2 microtubular protein, 2 microtubules, and always arranged at right angles to each other. Okay, 2 microtubules always arranged at right angles to each other and surrounded by the astral rays. What is their function? They form the spindle. They form the spindle and initiate cell division. They form spindle and initiate cell division. Meaning, if I talk about that cell, say suppose, I am talking about that cell which is in the process of cell division, then how will the structure be? This cell which is in the process of cell division will have a structure something like this, where at the poles of the cell, there will be the centrosome. How many proteins in the centrosome? 2. What is their name? Microtubule. Their name is microtubule. Microtubule, microfilament, these are the type of proteins. So microtubules, as of now, you'll have to remember. Then this will have such type of radiating rays like this. One more centrosome, 2 centrioles, and the type of protein. Like this is the. So this centrosome, can they divide? Yes, they also can divide. So one centrosome will form 2, occupy the 2 opposite ends of the cell. What are these 2 regions of the cell known as? Poles of the cell. Now that they have taken 2 opposite positions, they will result in the formation of a spindle-like structure. This is very much essential when the cell has to divide. This type of a structure is formed. What is this type of a structure known as? Spindle. And later in the 10th you will study that the chromosomes, they start getting attached here and start undergoing the process of division. They show the process of division. So this is a typical structure to show how cells divide. So just now I said, what is that centrosome and centriole? Centrosome and centriole is a cell organelle present only in the animal cells. They are not present in the plant cells and they are always meant for Formation of such type of a spindle during cell division. Clear? Let us do some questions very fast. Now, you just write down 10 questions, try answering. Wherever there is a doubt, you will tell. Give me the body. Okay, write down the questions. Neat, nice, you will write the questions and the answer. I will give you questions. After that, I will give you some time. You can fill it. Number 1. Name the following. Name the following. Sub number 1. Biologist who proposed that all cells arise from pre-existing cells. Biologists who propose that all cells arise from pre-existing cells. Biologists who propose that all cells arise from pre-existing cells. Give one line gap. Number 2. Do you know the answer? What is it? Rudolf Virchow. Right. Right. Did you write down the answer? Right. Number 2. Number 2. Why is protoplasm known as the living substance in a cell? Why is protoplasm known as the living substance in a cell? What will you write? What did I explain the other day? Why is protoplasm known as the living substance? What to write? The protoplasm contains variety of enzymes, proteins, which is necessary for the functioning of the cell. What is protoplasm? Protoplasm is what? Present way. The jelly-like substance inside the nucleus is nucleoplasm. Outside the nucleus all over is what? Cytoplasm. The cytoplasm and the nucleoplasm together, protoplasm. Write down the answer. Protoplasm is the jelly-like substance. Protoplasm is the jelly-like substance present inside the nucleus. Present inside the nucleus. And also between the nucleus and the cell membrane. Between the nucleus and the cell membrane. Full stop. The protoplasm contains, or it contains, variety of salts, ions, pigments, enzymes, proteins, etc. necessary for the cell functioning. necessary for the cell functioning, full stop. If the cytoplasm or nucleoplasm starts leaking, the cell will starts leaking, the cell will stop functioning. The cell will stop functioning. Number 3, question number 3. What are cisternae? What are cisternae? What are cisternae? What to write? They are, what are cisternae? They are the flattened, sacs, SACS. They are the flattened sacs or tubules or vesicles. Flattened sacs or tubules or vesicles present in the Golgi bodies. Present in the Golgi bodies. Present in the Golgi bodies. Next, why are lysosomes known as suicidal bags? Why are lysosomes known as suicidal bags? Why are lysosomes known as suicidal bags? Answer. When a cell is old or worn out, when a cell is old or worn out, when a cell is old or worn out, the lysosomal enzymes dissolve the cell parts. The lysosomal enzymes dissolve the cell parts and membranes. And throwing them out of the body as waste substances. At this time they are known as, or full stop. Due to this function, you write this way. Due to this function, they are known as suicidal bags. Yeah? Due to this function, they are known as suicidal bags. Okay, go to the next. What are chromosomes? Next question. What are chromosomes? Question mark. Next part of the same question, right? What are they made up of? What are chromosomes? What are they made up of? Answer. Chromosomes are coiled chromatin fibers. Chromosomes are coiled chromatin fibers present inside. Chromosomes are coiled chromatin fibers present inside the nucleus. Full stop. They are made up of DNA. That is, write on the full form. They are made up of DNA, that is deoxyribonucleic acid. Next, what are genes? Genes are functional segments of the DNA molecule. Full stop. They contain information. They contain information for encoding a protein, encoding. For encoding a protein that regulates, specific characteristic. Encoding a protein that regulates specific characteristic. Last, name the type of proteins present in centrosome. Name the type of proteins present in centrosome. Full stop. State their function or state its function. Name the type of protein present in centrosome. State its function. Answer. Proteins present in centrosome are microtubules, that form the centrioles. Full stop. Next slide. The centrioles help in formation of Centrioles help in formation of spindle. Formation of spindle during cell division. During cell division in animal cells. Cell division in animal cells. Okay, now just leave this page because I think animal cell organelle, what are we left with? Animal cell organelle, mitochondria only left. And apart from mitochondria, we have finished all. So animal cell organelle only mitochondria left. So this page, wherever you are writing down the answer, just leave this. Sure, you have got one more page left. Okay? Leave this page. For you, or which answer is this? Which answer is that you are writing? Which one?
Notice photo.
Why are you writing again? You did not write there? Achha. Now what you do is, leave this page. This page you leave, mitochondria we will do. And now in this page, fresh page, go to the next page. In this fresh page, you write down one question which you will complete as homework. Draw a neat, well-labeled diagram. Draw a neat, well-labeled diagram of plant cell. Draw a neat, well-labeled diagram of plant cell. Okay? The diagram will be more than half a page. Now, this is a ruled book, no? So you mark 13 lines, your diagram. Mark how much it is. Count, so keep a mark there that this much big my diagram has to be. So just put the mark. 12, 13 lines, definitely the diagram should be little bit towards the margin. With pencil only, complete the diagram, check from the textbook. In your textbook, the diagram is there, plant cell diagram you draw, complete and label with pencil. Now you complete that short exercise in your volume. Page number 12, go to that classwork exercise, small exercise you should be able to do. MCQ, only that one question we are not done, but never mind, we can complete that. Classwork exercise, see there, A sub number 1. A sub number 1 you do. This one. A sub number 1. Except for mitochondria you may leave also, or there is you may complete the answer, not the rest of the explanation and tell you the next lesson. So mitochondria only for animal organelle we are left. Next we will have to start with the plant cell. The first exercise only, number 1 only you do. Mitochondria, what can you write? What is the question there? Only the first one you complete now. Done? Done? Now go to those solved question answers. See there are some solved question answers. What is the first question?
It is said that the protoplasm cannot be analyzed chemically.
Take that question. What is next?
What is the difference between organ and organ?
Read this too. All these solved questions, these are all your text questions, huh? All are solved here. So keep reading this. Definitely you can expand, you can add your own points also, but this is standard, this much you have to write. Read the first 2-3 questions all from your text. See if you understand. If you have any doubt, you will clarify and ask. Read the first 2-3 questions already done. Read, see that you can have any complete. What is the third question? Read.
Do you think the cells of an elephant should be larger than the cells of a rat?
What do you think? Cells of an elephant, should they be big, large? No. Though the elephant is big, large, cells are not big, large. No.
Yes.
Cells will be only more in number. If the rat has got 1,000, elephants may have 10,000. Cell size, structure, shape, function will be the same. What is the fourth?
Differentiate between the following pairs of terms: protoplasm and cytoplasm, nucleolus, centrosomes, and chromosome.
Read those things. These are all your text questions. Read all. I'm not asking you to write separately, but off and all, I will give you short tests based on these questions. So read now, find out if it is understood. You write later. Now you just read and find out if it is understood. Writing work you can do on your own. Read and find out if it is understood. If there's any doubt, you can clarify right now. Tick all these questions, complete, nice, thoroughly you read them properly at home also. Next class when I discuss the chapter again, you should be able to tell me whether you are okay with it, if you have any doubts. Okay? And complete your work also, the diagram.